Hey guys, welcome back to the channel. So in this video we are going to discuss more about environmental biotechnology. So let's get started. So previously we have discussed about nitrogen cycle and I have given you an overview about nitrogen cycle and what it is all about. So let's get deep into the term of nitrogen cycle and what it is all about. So with this, so talk about the nitrogen cycle. So there are some very important bacteria that are involved in this process for making this process a successful one. So definitely let's just talk about some of the bacteria which are involved with the process of nitrogen cycle. So the first step or the first subheading that we have under nitrogen cycle is the biological nitrogen fixation. So leaving the term biological, so let's just concentrate now on the term of nitrogen fixation. So nitrogen fixation is generally conversion of atmospheric nitrogen to ammonia. All right, so which is known as nitrogen fixation. So how is it possible? So it is possible with the help of microbes definitely and some of the enzymes. So it's a definitely biological process and thus termed as biological nitrogen fixation. So let's talk about it. A reduction of free atmospheric nitrogen into ammonia in soil by living organisms is called biological nitrogen fixation. So I just said about what is nitrogen fixation. So definitely it is possible with the help of enzymes and microbes that are present inside it. So it is collectively termed as biological nitrogen fixation. So the molybdenum containing nitrogenase enzyme complex encoded by NIF gene complex is involved in the reduction of nitrogen to ammonia is present exclusively in prokaryotes. So definitely this conversion is helpful or happens with the help of the enzyme known as nitrogenase, alright, and which is encoded with a gene known as NIF gene or NIF gene complex, alright, which helps in the reduction of nitrogen to ammonia, alright. Also, this is extremely energy expensive reaction that involves splitting of nitrogen gas molecules and adding hydrogen to make ammonia. So definitely splitting nitrogen atoms is a very costly process because it's bonded by triple bonds. Alright, so splitting triple bonds requires expenditure of a lot of energy. Alright, so definitely we need to split it with giving amount of energy such as 669 kilocalories to split the nitrogen atom. Alright, so we get two atoms of nitrogen here. With that we can get ammonia. Alright, so this is how the process gets carried out, and while whole process requires 8 nitrogen and 16 ATP. So this is the important point overall in this nitrogen fixation. So the conversion of atmospheric nitrogen to ammonia involves or requires around 8 electrons and 16 ATP molecules for breakage and formation of ammonia. Alright, so this is the net figure that we have is the 8 electrons and 16 ATP molecules. Moving on with this. So talking about the biological nitrogen fixation in its broader terms. So how can it be done in different ways and different microbes that are present in the soil? So nitrogen fixing microbes can be of two types. So we just talked about the enzyme which is involved in the process is the nitrogenase, alright, which is coded with NIF gene complex. So talking about some of the microbes that are involved in which can be three types, which are symbiotic, free living and photosynthetic nitrogen fixers. So symbiotic counts under symbiotic microbes can be Rhizobium species. Free living microbes can be Azobacter, Nostoc and Pseudomonas species. Also we have photosynthetic nitrogen fixers which can be cyanobacteria. And these are genetically engineered biological nitrogen fixation to replace synthetic fertilizer completely. So definitely this is a point that we are looking in our near future for if they are possible, if the microbes can be genetically engineered so that these nitrogen fixation can be possible, and thus we can replace the synthetic fertilizers completely. Alright, so synthetic fertilizers definitely devoids the soil and degrades the soil quality. So definitely genetically engineered all of these fixation microbes can be used to utilize the process. So moving on. So talking about the symbiotic nitrogen fixation, so which is the first topic. So we just went through that there are three types of microbes that can occur. Out of the three, the first was symbiotic, second was free living, and third was photosynthetic. So talk about the first one, which is this symbiotic nitrogen fixation. So symbiotic nitrogen fixation or symbiotic microbes, one of the example that we just right now studied is the Rhizobium species. So we're going to study about that type, and we have a picture corresponding to that as well. So Rhizobium is the most important nitrogen fixing bacteria, and Rhizobium stays in symbiotic relationship on the roots of legumes. Also, high level of nitrogen fixation catalyzed by the presence of molybdenum containing nitrogenase. So definitely this microbe Rhizobium helps in the process of nitrogen fixation. Alright, and these are present in the root nodules or roots of legumes such as clover, soybean, peas and alfalfa. Our high level of nitrogen fixation is catalyzed. It is definitely the process is enhanced with the help of the enzyme known as nitrogenase. Also, nitrogenase inhibited by ammonia and also susceptible to oxidation, hence red pigment like hemoglobin present in root molecules absorb oxygen, thereby limiting oxygen supply. So nitrogenase, or which is inhibited by ammonia, also susceptible to oxidation, hence we have this term known as like hemoglobin that is present in the root nodules as well, which helps the root nodules that absorb oxygen from the soil and other nutrients, so thereby limiting oxygen supply. So definitely due to presence of like hemoglobin, this makes the entire oxygen content less because all of it gets absorbed here with the help of like a hemoglobin in the root nodules. So definitely oxygen content is always a limiting term or a limiting quantity. Also, plant provides carbohydrates and oxygen to the bacteroids for cellular respiration, as nitrogen fixation bacteria are aerobic. So nitrogen fixation bacteria, so all of these microbes that we are studying about are aerobic in nature, and definitely not they are facultative, nor they are facultative anaerobes or anaerobes. So definitely simply they are aerobes or aerobic in nature, all of these microbes. So talking about the Rhizobium legume symbiosis, or these nitrogen fixation carried out by Rhizobium species in the presence of nitrogenase. Alright. So bacteria gets protection and organic nutrients. Legume gets a supply of nitrogen for growth. Definitely also presence of leg hemoglobin is used. Leg hemoglobin is present for providing them proper oxygen. So as you can see a picture here, a lot of pictures here. So let's just talk about this picture here first. So this is the root, these are the root nodules and a lot of the stuff here. This is one more root nodule that we have. This is the enlarged form of root nodule. So this is all we have marked as like 1, 2, 3, 4. So let's just go through that. So this is the first part. So this is the one. This is just a normal plant. And talking about enlarging this part, in this we have this, all of the hair roots and all of the stuff, and 60% of terrestrial nitrogen fixation things are there, and rhizobium attached to root cells. So moving to the second point, all of the rhizobium which are attached to this. Alright, and an infection thread is formed through which bacteria enters root cells. So definitely rhizobium enters the roots so that it do the job. An infection occurs, and thus bacteria change into bacteroids. So bacteria change their forms. These are enlarged here, change into bacteroids, and packed root cells enlarge, and definitely enlarged root cells form a nodules. Alright, so this is a clear picture that we are getting. So these packed root cells, all these packed root cells lead to formation of nodules, or enlarged root cells form a nodules, as you can see here. Alright, so moving on from here. So this is another picture that we have. This is a raw picture that I got it. Alright, so these are all the rhizobiums that are present and which enter the cells and thus forms packets or root packs, or which lead to the formation of root nodules. Alright, also root hair surfaces, root hair surface lectins especially bind recadhesin knot factor proteins induce root hair curling. So definitely presence of this factor already, hecadhesin knot factor protein induce root hair curling. So as you can see here, so knot factor, due to the presence of knot factor of recadhesin, It induces root curling and root hair deformation and bacterial attachment by recadicin and host lectins. So definitely it attaches the what you call rhizobium inside it due to the presence of knot factor. Alright, so this induces root curling and so that the rhizobium gets trapped inside it and thus enter the root cells. So moving on with this, so we have another perfect picture of here, as you can see here. So this is curling of root which traps in the rhizobium microbes and which thus enters the cells and thus infect the cells with the rhizobium, and these cells form packets and or what all bacteroids we can say. These form bacteroids in the cells and these leads to formation of root nodules. Alright, so rhizobium stimulates the formation of an infection thread through that the bacterium moves into and grows within. Ultimately, infection is within plant root cortex cells where rhizobium transforms to a non-motile bacteroid state covered in host membrane. So these form bacteroids, or these, as you can see, so these are single cells. So these lead to formation of packets or pre-bacteroid membranes and thus lead to bacteroids which are covered in a host membrane. Alright, so these are covered in a host membrane as they move forward in the process, and transformation continues to form symbiosome wherein the bacterium is now pleomorphic and begins fixing nitrogen. Alright, so these transformation continue to form these structures which are known as symbiosome. Alright, wherein the bacterium, but these are the bacterium, are now pleomorphic in nature and begins fixing the atmospheric nitrogen to ammonia. Alright, also leg hemoglobin controls the oxygen supply to the bacteroid. So leg hemoglobin plays an important role in the oxygen supply. So talking about the last thing about in this video that we have the table, which is the characteristics of the genera Rhizobium, Bradyrhizobium and Azorhizobium. So it's a comparison with other related legume nodulating bacteria. So here we have another lots of features, lots of rhizobium species and all of the stuff here, right? So these are flagella on liquid medium and solid medium. We have growth on nitrogen fixed outside of plants, growth rate in culture, location of nod and nymph genes, host specificity rate and agriculture significance. Alright, so nod and nymph genes we have just studied about. So nod is the gene that helps in the root curling with the help of radicin, and nymph is present with the enzyme which is nitrogenase that helps in the nitrogen fixation of nitrogen to ammonia. So let's just keep this video till here. I'll be back with another video very soon, so stay tuned and thank you for watching this video.